This presentation is really a presentation to give updates in terms of research and clinical trials and the registry. And Pat and I have decided that for each of the CMD subtypes, we will provide these updates every 6 months so that our community can start to have a better understanding of what is actually going on and are better informed.
Hey, this is Patrick May. I'm along with Anna and Rich Cloud. Kind of got together and several years ago and helped co-found Cure CMD. Been involved ever since. Currently serving in the chairman role, and have a daughter, Aubrey, who is 8 years old and is Merosin Deficient CMD. And you know, like everybody else, kind of looking at all of the ways to kind of help push all the science and everything forward, and that's kind of how everything from our end kind of got started. And I guess that's it.
Great, thank you, Pat. And I'm Anna Rutkowski. I live in Los Angeles. I have a 16-year-old daughter who has CMD, a different form of CMD. And together with Pat and Rich originally co-founded Cure CMD. So in today's presentation, I'm just going to touch very briefly upon Cure CMD, and then talk about the CMDIR, which is the International Registry, then provide updates in terms of current clinical trials, and also provide updates on research that is currently underway. So this is just a picture of us running to raise money to fund research. As many of you know, Cure CMD has an annual research grant program, and we fund research every year. Over the last 6 years, we have funded over $1 million in research grants. Those are international research grants, so we have funded people in France, in England, and throughout the U.S. In order to move forward the best science for CMD. Cure CMD was founded in April of 2008 by 3 parents. And it is an all-volunteer organization, meaning that none of us receives a salary, except for one person who is part-time, and that is Rachel Alvarez, who is the Associate Director of our registry. Cure CMD's mission is to raise CMD awareness and identify treatments, and really our vision is that one day all children and adults will have an opportunity to participate in a clinical trial. So over the last 6 years, we've really built, according to a project plan, the infrastructure to support science, have built a growing community of scientists, clinicians, families, government officials, have focused on building a drug pipeline and identifying the barriers to getting to clinical trials, whether it is natural history studies, outcome measures, or adverse event rates. So we really believe that the key to getting to clinical trials, in part, is to have a registry, a database, a place where the international community, families with CMD, can put in their information so that we can track what problems arise in CMD to find out how many people there are who are affected, how is the condition, the disease affecting them, and what kind of care are they receiving. This is very, very important because when I speak with pharmaceutical companies who are interested in these diseases, the first question they ask me is, how many people are there in the world with this condition? And often I do not have a good answer for them. In fact, I never have a good answer for them because we really don't have a good number, a prevalence number of how many people are affected. And that is in part because there are still many people across the world who do not have a diagnosis. So currently in the CMDIR, which is the Congenital Muscle Disease International Registry, this was a registry that we launched in 2009, and it has since been expanded to include not only congenital muscular dystrophy, but also congenital myopathy and congenital myasthenic syndrome. This is the website, www.cmdir.org. It is currently available in Spanish and Portuguese, and Chinese is being launched in the next couple, that should be finished by the end of the month. We are working on French, and the idea is to have families put in their information, also adult affected individuals, so that we can start to have a better sense of how many people are affected across the world. There are currently 1,500 registrants from 68 different countries, and we will break that down further in this presentation. So the purpose of this registry is really has many purposes. You can see that there is the figure of Bart Simpson, who is looking kind of confused, and many times families are a little bit confused. They may not have an official diagnosis. They may just have been told that their child has a congenital muscular dystrophy. We would like everybody to register, even people who don't have an official genetic diagnosis, may have a muscle biopsy diagnosis, or may have just been told that they fall into this group of conditions. We also ask families to register who have lost a child to one of these conditions. And the purpose of the registry is first to provide help and support to families. So when you register, you will receive an email with multiple links to various support groups. If there is a clinical study or trial that you fall into in terms of eligibility, there is a counselor who will reach out to you. And then we collect medical records on all people who are registered. And the reason to collect these medical records is for us to learn more about these diseases. We would like to know, are there underreported complications? Are there people in certain countries who are living longer because they are getting a certain type of management? And for example, how soon do people with any given condition, such as LAMA2, CMD, or Merosin Deficient, need to go on breathing support at night? These are questions that we can start to answer by having these medical records. So we curate the medical records, and for everybody who is registered who has medical records that have been sent in, we trend the key values like the weight of a child with LAMA2 over time, and the weight is extracted from the medical records. This allows every family to start to trend how their child is doing on some very key variables: weight, breathing function, heart function, scoliosis over time. And then we ask that people come back on an annual basis in order to provide us with an update on how their child is doing. So everybody who registers puts in their contact information, and then there is a detailed consent that explains to everybody who's registered what we do with their information. And then there is an 80-question intake survey, which you will see not everybody has completed. Starting in January, there will be an annual survey of 25 questions, so we can see how often somebody has been hospitalized in the last year, whether they're taking any medications, how their breathing function is doing over time. In addition, this database, the CMDIR, has links to a biobank where we store patient cell lines and a tissue repository where we store muscle biopsies, so that there can be information that is shared with those specimens. That information is anonymous and does not identify the individual. So here you see the breakdown in terms of registrants per country, and across the top you see November 2013 to August 2014. You can see that the majority of registrants come from English-speaking countries, and that is in part because the registry was first launched in English. But now that it has been translated, we are hoping that we start to see increased registration from countries where English is not the primary language. In a rare condition, it is very important to register people from all over the world, because there is no chance that you can go to a clinical trial with just the people from one country. It is too rare of a condition in order to have enough people to participate without bringing on board people from other countries. And that is in part why this registry is international. We really truly have to find everybody worldwide. So you can see that the numbers are slowly growing. This is very positive, but this is still a very small number when we think about the total number of people who are affected with congenital muscular dystrophy and congenital myopathy and congenital myasthenic syndrome. So here is the breakdown of the people with different conditions, okay? And you can see that for LAMA2 CMD, or Merosin Deficient CMD, as it is also known, you can see that the numbers, the total numbers registered is currently 170. So this is a very small number. You can imagine when a pharmaceutical company comes to me and says, oh, we've got a product that we're very interested in moving forward, how many people do you have registered with LAMA2 CMD? I have to say we have 170, but we know that this is not the true reflection of the worldwide population. And that is why it is so critical for us to find everybody who has these diseases and to ask them to register.
So, Anne, it's 170 in the entire world?
So currently we have 170 people who are registered from 68 different countries.
Right.
We know that there are many more with LAMA2 CMD, but we know they just are not registered. One, they don't have a diagnosis. Two, they don't know about the existence of the registry. Three, they are worried about putting their information online. So there are many barriers to getting people to register, but one of the most significant barriers is the fact that people do not have a diagnosis, and I'll talk a little bit more about that in a moment. You can see, though, that over the last basically 9 months, there has been a slow but steady increase in numbers for all subtypes, and that's really fantastic. It means that slowly people are starting to see that they do need to register, but it really also means that all of you need to go back to your neurologist, or if you are in fact a neurologist, ask your patients to personally register, because this is the only way that we will build up enough numbers to be able to move forward into clinical trials. This is to give you a sense of how complete people's information is in the database. For those of you who deal in information technology, you will know that a database is only as good as the information contained within it. You can see that out of the over 1,500 people who have registered, most people have entered their demographic information, which is their contact information and their address. A little more than half have also completed the 80-question intake survey, which is, we would love to see that number be much higher, because that intake survey provides us with information about how that child or adult is currently doing. 221 people who are registered originated from a traveling clinic that I run together with Dr. Carson Bonneman. And we will be in Buenos Aires, which we are very excited about, in December, and then we'll be in Atlanta in the United States next January, and then in Calgary, Canada next summer. And this traveling clinic system came about because we recognized that many patients do not have access to genetic testing, or they may not carry a diagnosis. So the purpose of this clinic was to travel around the world and work together with local physician teams, neurologists and physiatrists, to see people who do not currently have a diagnosis and to provide assistance with access to genetic testing. Okay? And you can see that there are a large number of people in the CMDIR who have never had a genetic test or have not had any positive results from the genetic testing that has been ordered. So again, in order to participate in a clinical trial, you need to be genetically tested in order to know that for sure you have this condition. In many cases and in countries, we stop at a muscle biopsy, which may give an accurate diagnosis, but typically for clinical trials, a genetic diagnosis will be needed. So what is your contribution? And I'm talking to everybody on the phone who has an affected child or is an affected individual. Really, the only way that we as a community can achieve progress is for each of us to contribute. And your contribution to the registry is to sign a medical release, or if you live in a country that does not allow the release of records, to obtain your own medical records and to send them in. We also ask that everybody take an annual survey, which will take about 15 minutes starting in January. And if you've not already completed the intake survey, to go back to the CMDIR, complete your intake survey, and send in your records. All records that are sent in are attached to your profile and will then be curated for you. You will have access to all of your records wherever you have online access. And so it becomes like your own patient chart, your own medical chart in the registry. We also ask for those who are interested, if they are interested in donating blood or skin cells to the biobank, which is located in New Jersey in the United States, to consider a donation. Many times in the U.S. we have been fortunate that several neurologists have been willing to send some of the skin cell lines that they've developed to the biobank. And this biobank is now actively being used by scientists around the world who are requesting cell lines from people with CMD. So it's been a very big success. The tissue bank, which is also located in the United States, is a place where people can have their muscle biopsy sent to. So if there's no doubt about your diagnosis and your muscle biopsy is simply sitting in a freezer somewhere and not being used for active research, this is a way that you could ask that your muscle biopsy be sent to the tissue bank, where it then becomes available to scientists internationally who are conducting research and allows them to learn more about our conditions. If you have any questions about the biobank, the tissue bank, or the registry, you can always contact counselor@cmdir.org. And you can do this in any language, and we will use Google Translate to figure out what the issue is. We also have, fortunately, several people in our office who speak French and Spanish and German. So at least in those languages we can provide some additional assistance. So some people have said to me, well, how does it matter, why does it matter that I participate in a database? And what I really would like to make clear to everybody is that a database is the way that we can slowly start to change care and management and improve management of CMD. So what I am showing you here is a paper that is in the process of getting published by Dr. Jim Collins, and it drew on data from the registry as well as 6 other centers. And this is data from several people. I think it was over 54 individuals with LAMA2 CMD. And what he was able to show is he looked at the breathing, so their forced vital capacity. This is the test that is performed on an annual basis to check people's breathing function. And what he was able to show in the red line is people who are walking with LAMA2 CMD do not have a drop or a decline in their breathing function over time. On the bottom axis is age in years. But kids with LAMA2 CMD who are not walking, both those who are not on ventilation, which is the top line, and the green line are those that are on ventilation, have an annual rate of decline in their breathing function over time. Now you're going to say, well, I understand that this is important to know that in terms of preparing my child's doctors to be aware of a change in breathing function over time. This also provides us with an opportunity in future clinical trials to use breathing function as a measure of global muscle strength, so that we could perhaps give a drug, and instead of testing whether or not that drug improves arm function, we can say, does it improve breathing function?
Anne, can I ask you a question?
Yes, go for it.
The red line, you said that those are kids who walk and so they don't have any decline in their breathing, right?
That is correct. So
yeah, go ahead.
So what he found is he clustered the kids who are non-walkers, who do not achieve the ability to walk, and he clustered the kids who are walkers. And what he noticed is the kids who are able to walk do not have a change in their breathing function over time. But the kids who do not walk have a decline in their breathing function over time. And the difference between the blue and the green line is the blue line are kids who are not currently on breathing support. They typically have a slightly higher forced vital capacity. And the kids who are on breathing support start with a lower forced vital capacity. It makes kind of logical sense that doctors would prescribe breathing support for kids who have a lower forced vital capacity. So, but in both cases, regardless of whether or not you were on breathing support or not on breathing support, you did have a decline. Right. Interestingly enough, the decline was steeper for those who were not on breathing support.
Okay, and the kids who could walk, could they walk, like, maybe it's a silly question, but could they walk like on their own, or did they consider like those kids that had like walking aids or assistance to walk?
That's a very good question, and it is not a silly question at all, because these are always the types of questions that we need to ask ourselves when we divide kids into different groups to look statistically at differences. And for this purpose of this one, it was kids who are able to walk unassisted.
Unassisted, okay.
Can I ask a question?
Yes, go for it.
But I wanted to ask, are there a lot of kids who are able to walk with
LAMA2 CMD? No, actually, in fact, the majority of kids with LAMA2 CMD and adults with LAMA2 CMD have not achieved the ability to walk. So it is a very small percentage that achieve the ability to walk, and these kids can present later on, so they may not be perceptibly weak directly at birth. In the same family, you can have two kids, one who maintains walking ability and the other who maybe had walking ability for a very short time but loses it. So there's always kind of there can be a difference in degree of severity, but classically, typically, LAMA2 CMD children never achieve the ability to walk. So they are non-walkers from birth forward. And you can really understand the importance then of having one central location from which to then pull data from multiple different countries. Because you can also imagine that the approach to breathing care may be different in different countries, and therefore it's very important to be able to assess how things change over time from multiple different countries in kids who are genetically confirmed with LAMA2 CMD. Okay, so I'm going to talk to you about several studies and trials And I'm first going to talk to you about the studies and trials that are currently ongoing, and then speak very briefly about two that were completed, sorry, it should say completed in 2014 instead of 2015, that were completed this year. So we are starting to conduct studies, as I had mentioned, through the registry. And I wanted to provide an example of this because it really illustrates the power of the registry and the database. So this is a study that we launched called CMD Sleep. And the purpose of this study is to look at the data that we have in the database and determine at what age sleep studies are first abnormal in children with CMD. And then to look at, in those kids who have an abnormal sleep study, if they have had pulmonary function testing, what their forced vital capacity was at that time. So you're going to say, well, why is this important? And the reason that this type of a study is important is because current breathing care is very variable. Even in any country, it depends on which doctor you see, depends on whether you are told how often to get your breathing function assessed, and what interventions are provided to you. We already know, and you saw that now in that graph, that breathing changes over time and that breathing declines over time in kids and adults who do not achieve walking. And so what is really at the core of this is that breathing issues drive quality of life and survival in LAMA2 CMD. So figuring out at what age these sleep studies become abnormal and how this is related to pulmonary function testing can actually help provide guidance and improve medical management over time and make it more standard. So how can you contribute? Again, part of this presentation is to provide you and empower you with the ability to step forward and contribute, because as we've already said, Cure CMD is all volunteer. We contribute on a volunteer basis, and the only way that we can move forward as a community and achieve change, get to clinical trials, is if everybody steps up. So if your child or you as an affected individual has had one or more sleep studies and or pulmonary function tests, we would be very grateful if you could send in a copy of these reports to the CMDIR, and also to keep a copy of those reports in your house in a folder which you can track over time.
Anne?
Yes.
I was wondering at what age is it normal to start doing like annual studies? Or what age should you start?
Great question. So the CMD consensus guidelines, which was a document that was put together involving 80 experts from around the world, basically stated that in children who are walking, once they lose the ability to walk, they need to have a sleep study. And then it was left somewhat open. Basically what the recommendation is that starting at age 5, every child should start to have annual pulmonary function testing. And if that pulmonary function testing is abnormal, the forced vital capacity, then it should be done every 6 months. Now, from Duchenne muscular dystrophy, there was a larger study that showed that once a person has a forced vital capacity of 60%, they may start to have sleep disordered breathing. So the recommendation is really once you have a forced vital capacity of 60% or less, that you get a sleep study. If that sleep study is normal, but the forced vital capacity is abnormal or is low, then it makes sense to follow that individual on an annual basis with sleep studies. The big problem is that it is not clear at what point people should receive breathing support based upon their sleep study. So a sleep study can be abnormal for something we call sleep disordered breathing, or could be abnormal for something we call hypoventilation. And pulmonologists themselves are not always on the same page in terms of when they think that a non-invasive ventilation should be initiated. The other thing that we know in LAMA2 CMD in particular, from a large study that was published on 51 individuals out of the UK, is that there appear to be 2 time periods where kids with LAMA2 CMD require breathing support. And that is between age 0 and 5. So some children are really quite weak at birth, and they require breathing support within the first year of life, and then they need to be on breathing support. And then the next wave of when kids need to start breathing support is typically somewhere between age 11 and 15. So really it's a somewhat of a vague recommendation, in part because we don't have enough data to know when sleep studies become abnormal. I organized a meeting with 3 other pulmonologists in the Netherlands in last December, and that meeting report will be published in the next 2 to 3 months. And that recommendation is a much stricter recommendation, and the recommendation will be that every child with CMD receive a sleep study at the time of diagnosis. So let's say you are 6 months old and you receive a diagnosis of LAMA2 CMD. The recommendation is now going to be that you get a baseline sleep study at that first time of diagnosis to make sure that at that early age there are in fact no sleep abnormalities that would require an intervention. If that sleep study is normal, then unless the person with LAMA2 CMD, the child, has multiple hospitalizations or frequent chest infections or failure to thrive, meaning they don't put on weight, those are all signs that there may actually be an abnormality in breathing, and there may be a need for non-invasive ventilation. So if a child basically passes that first sleep study and doesn't require hospitalization, doesn't have frequent chest infections, and is able to gain weight, then starting at age 5, you get your first pulmonary function test. And then based on the pulmonary function test, you use that as a guide to determine whether or not a sleep study is needed.
Can I ask a question?
Yeah.
Are those the only sign, like, are there any other signs that you would have to know that if they had a problem with it?
So problems with breathing issues are very gradual in their onset. In little kids, so I'm speaking under age 5, no child is going to tell you, I feel short of breath. So typically the main signs are not able to gain weight, frequent chest infections, and frequent hospitalization for chest infections.
What if there's no infections or no, like they don't gain weight that much, but there's none of the other stuff?
Yeah, it's a tough question to answer. I think what is needed is it needs to be a multidisciplinary approach where you meet with a nutritionist, you look at hydration and constipation. Constipation is a big driver for no weight gain in LAMA2 CMD. So adequate management of hydration, constipation, and then most kids are very chatty, they're somewhat picky eaters, and they will not eat very much at the table. So it then means that you have to sit for an hour, and I always say that CMD is many things, but one of the best things it is an antidote to a fast-paced life, which we are all under pressure to join in. And so you have to slow down and really sit and eat for an hour. There have to be multiple snack times, and if the child is starting to go to school, toileting needs to be really well worked out so that the child doesn't feel awkward and does not drink because they are worried about having to be brought to the toilet, and that is different from other children. If all of those things are done and the child is still not gaining weight, then it is possible to say, could we get a sleep study just to make sure that this is not the issue? And then even if it is slightly abnormal but does not warrant at that point some ventilatory support, you now have a baseline from which you can go to say, how does the sleep study change over time?
Okay.
A sleep study is also not a big picnic. I mean, often you have to go into the hospital and then you get lots of electrodes placed on you, and you have to have something put up in your nose just to measure the carbon dioxide. It's not horrible, but it's not the most comfortable experience. Most people don't sleep very well when they sleep for their sleep study. One of the things that would be great to see a big change, they've started in adults to do sleep studies at home. But it's not clear that everybody endorses that approach in children. You know, that's something to keep in mind as well. A sleep study, like any medical test, is not perfect, and unless you're really able to fall asleep in the hospital, you may not get a fantastically accurate result.
Mm-hmm.
It is still worthwhile to do them because it really will give you a baseline sense of whether problems that you're encountering in the management are related to breathing issues. Any other questions? And then the one last point is a sleep study result is best interpreted by a pediatric pulmonologist who has experience in neuromuscular and experience in young children with a neuromuscular condition, or a physiatrist who has experience in neuromuscular. Most pediatric pulmonologists are very comfortable treating asthma, but don't necessarily have neuromuscular expertise, and their ability to interpret the sleep study may not be the same as somebody who has a lot of expertise. So we're going to talk now about the hyperinsufflation trial. So this is a trial that was started in November of 2013. It is a trial that came about because of my traveling and meeting Dr. Susanna Quijano-Roy in France, who had decided all on her own that she was going to do something to try and treat the annual rate of change, the annual decline in breathing function in her patients with CMD. And so she decided to start all of her CMD patients on a daily breathing exercise using a machine in France, and was able to show in her patients that their breathing function stabilized, and in some patients it improved. So I came back to the U.S. and decided that this was really fantastic. The idea behind this is that people with CMD develop contractures in their chest wall, just like you have contractures in your arms, your legs, your neck, your ankles, and that these contractures are leading to chest wall stiffness, and that this is in part responsible for the annual decline in breathing function. So her idea was, if we fill somebody's lungs with air and thus stretch out the chest wall from the inside out, might that actually prevent this decline in breathing function? And most people, when you explain it this way, it makes sense to them that this is something that you might want to do. So we decided in the United States, Cure CMD, working together with a group of pulmonologists, pediatric pulmonologists from Cincinnati Children's, Dr. Hemant Sawani and Dr. Raouf Amin, and Dr. Hank Meyer from the Children's Hospital of Philadelphia, that we would launch a clinical trial to determine whether daily breathing exercises stabilizes breathing function in LAMA2 CMD and in collagen 6 CMD. So currently we have 30 individuals who are enrolled in this trial. Our target is 35 individuals, so we actually need 5 more individuals enrolled by December of this year. Otherwise, this somewhat invalidates the trial because we need that number in order for this to be statistically significant. And people who are eligible are kids who live in the U.S. and Canada between the ages of 5 and 20 years who have a genetically proven or muscle biopsy proven LAMA2 CMD or collagen 6. And for those individuals with LAMA2 CMD, they cannot currently be on BiPAP, and in order to be eligible, must not have achieved walking and have a forced vital capacity between 30 and 80%. So I have been working on recruitment for this trial since November of last year. I've reached out to over 120 families, and we've enrolled only 30. So one thing that becomes very clear is in rare conditions, everybody really needs to step forward in order to participate for a future clinical trial, because even though it is an inconvenience to participate in a clinical trial, and there are clearly reasons that people don't participate, we won't make our goals and our targets for enrollment in rare conditions such as LAMA2 CMD without having the entire community step forward. The next study, which is not officially launched yet, but will be launched, we are anticipating in the middle of November, is the Omigapil Pharmacokinetic Study. And the goal of this study is to find the best dose of Omigapil in CMD for a future definitive clinical trial. So this is not the study to determine whether or not Omigapil works in CMD. It is the study to find which dose to use for that future clinical trial. Omigapil, why are we interested in this study and in using Omigapil? The reason we are interested is because Omigapil has shown a positive effect in 2 different mouse models of LAMA2 CMD. It's a drug that blocks the signal for muscle cell death, which is called apoptosis. And to get to a definitive trial, we actually need to know what dose to give. This drug has already been used in 2 adult human trials in neurodegenerative conditions, and in those conditions did not show an adverse event, but also didn't show a benefit. And so it wasn't until a drug company came along, Santhera, to license the drug and to perform some studies in CMD mouse models that we were able to then move this forward as a potential for a future clinical trial. So again, how can you contribute? Do you have a question? Okay. If you live in the U.S. or Canada and have a child between 5 and 16 years of age who, again, is genetically or muscle biopsy confirmed with a forced vital capacity between 30 and 80%, your child may be eligible to participate in this pharmacokinetic study. And as soon as the study is officially announced, we will be launching information through the registry about whom to contact in order to determine whether you are eligible and to then enroll in this pharmacokinetic study.
And I have another question.
Yes.
I was wondering, why is the age 5 years old? Is it because of like the speech of the kids? They can tell you what's going on, or like what is the age? Like why is that the requirement?
It's the reason for the age is because at age 5 you can start to get accurate pulmonary function testing in kids. So this study is going to measure breathing function, muscle strength testing, quality of life in the individuals who will all be treated with the drug. And the reason to do that is we are looking for a signal to determine whether the drug seems to show a positive trend or benefit even in a short-term pharmacokinetic study. Because if we are able to show a positive benefit, this will make it much easier to bring on board investors who we will need to bring on board for the actual definitive clinical trial, which will cost millions. Once this drug goes through a definitive clinical trial, and if it in fact shows that it is showing a benefit in CMD, then the drug will be made available for all children with CMD, regardless of age, as well as adults.
How long will this whole process take?
So this particular pharmacokinetic study is a study that will last 27 weeks. So it's basically 7 months participation. And the way that the study is set up is that participants will enroll in a staggered fashion. So the pharmacokinetic study we anticipate will take probably about a year to complete, and will then take about another a year to analyze the data. At that point, once the year and a half is complete, the study is complete, the data is analyzed, then we can decide whether we can bring on board investors to launch a definitive clinical trial. Most definitive clinical trials are set up where half of the kids are randomly selected to not get the drug and half the kids are randomly selected to get the drug. And then one determines whether there is a difference in motor function, breathing function over time during that study. There are other ways you can design the study, and that would still need to be decided. The actual definitive clinical trial typically runs somewhere between 12 and 18 months, and then you have to analyze the data. So you can see that this entire process is still going to take anywhere between 2 and 5 years. And one of the biggest hurdles will actually be to bring on board additional investors to fund the clinical trial.
Hello, Annie, Cecilia. I'm sorry, I couldn't introduce myself earlier. I'm calling from Barcelona.
Yes, it's great to have you on the call.
Thank you. I'm very pleased to join you. I was just wondering, this initial pharmacokinetic study, will it involve muscle biopsies before and after the treatment to look at changes or other biochemical outcome measure to see if the apoptosis rate is changing with the treatment?
Yeah, we decided that for the pharmacokinetic study that we would not be that invasive. So there will not be muscle biopsies performed on the kids before and after. You know, I think part of the challenge always is to define which muscle is most likely and in which part to show apoptosis. And it's unclear whether any biopsy that you obtain is really going to give you the same yield as you would have received if you had biopsied in a different location. So I think that's always the big challenge with muscle biopsies. And in addition, you know, it's interesting when you look at a publication from Professor Hayashi in Japan, where she compared the muscle biopsies from 10 individuals with LAMA2 CMD, 10 with other CMD, and 10 with dystrophin, with Duchenne. And what she showed is that the apoptotic signal measured by caspase was very high in the first 0 to 1.5 years of life. But if the muscle biopsies were obtained after that time period, that the signal actually diminished.
Yes, I think that's our experience as well. It's very variable between patients, between the biopsies.
Exactly.
Okay, thank you very much.
Yeah, no problem. And it's a great question. And given that that's the target, you would it would be even better if we had a serum signal that we
could.
Yes. But unfortunately, we don't have one. So that's a challenge, though.
Okay, thanks.
Yes, absolutely. Okay, so in terms of completed studies, the first study that I want to mention is this comparative outcome measure study, which was a 5-year natural history study that Cure CMD led in conjunction with the NIH, with Karsten Bonneman and Katie Mayer, who have been wonderful collaborators, as well as Nina Jane at the NIH. And this is a study that went on for 5 years, where the same families with LAMA2 CMD and collagen 6 came back every summer to undergo many different tests, including tests of breathing function, muscle strength, and quality of life. And the reason that we invested this time is that we needed to determine how quickly things change in skin, whether they change at the same rate for everybody, in order to prepare for a future clinical trial to say we can use, for example, breathing function, or there's a test called the MFM32 that was developed in France to assess muscle strength. And what we wanted to do was to determine what endpoint we should use for future clinical trials and how quickly that changes and how consistently it changes in LAMA2 CMD and in collagen 6 CMD. So we have results from this 5-year study. The data from the first 2 years has been analyzed and has been published. That is the publication you see below. And what that basically told us is that this MFM32, which is a global test of muscle strength, is really the gold standard to assess muscle strength on an annual basis. And now the data from the last 3 years is currently being analyzed. We anticipate that this study will really provide a lot of additional data on care and management, and so there will be several other publications that will come out of this 5-year natural history study. We launched a separate effort, again, through the CMDIR called CMD Breathe, and this was a 2-part study to evaluate current breathing care in the U.S. And we had 103 pediatric pulmonologists respond to a survey to provide us with insight on the care they currently provide to kids with neuromuscular disorders, and then interviewed 140 CMDIR registrants to provide insight in the care that they have received in the last 5 years for their child. So we think that this will help us understand whether, in fact, care is variable and whether variability in care is problematic. And with this, we hope to push for greater standardization in care for people with CMD. So now I'm going to shift gears and talk about 3 scientists who Cure CMD has funded over the last years. Highlight some of their research findings, and provide you with insight in how Cure CMD has used the funds raised by parents and affected individuals like yourselves to be able to actually create these annual grants. So the first person I would like to highlight is Dr. Sweta Gergenrath at Boston University. And Dr. Gergenrath has been working in the mouse model for LAMA2 CMD for many years. The mouse model for LAMA2 CMD, there are several, but the one that is most commonly used is called the DYW mouse model. So if I refer to that, that is what I'm talking about. I'm talking about the mouse model for LAMA2 CMD. So we initially funded her to look at the role of IGF in combination and without BAX inhibition. So this was a study to really look at the role of the inflammatory processes and various signaling pathways that are abnormal in the LAMA2 CMD mouse model. And what she was able to show in this initial study is that when you combined a genetic strategy to inhibit BAX signaling and you provided IGF, you increased IGF, that the mice had improved survival, improved weight, and an improvement in muscle strength greater than with either treatment alone. And this was really the first work that highlighted this idea and concept that in LAMA2 CMD, combination therapy is likely going to be the way of the future. So I always compare this to HIV, which is a disease where one needs to be on multiple combinations of medications in order to optimize health. And this is the future that I anticipate for LAMA2 CMD, that kids and adults will be on varying combinations of drugs in order to optimize their muscle strength, their breathing function, and their overall survival. So the next study that was funded through Dr. Gergenrath is to evaluate breathing issues in this mouse model. So nobody really understands why this mouse model dies. It is a pretty severely affected mouse model, and as such is really a very good mouse model of the human disease. So the mice develop changes in the muscle that look very similar to the human changes in the muscle. They develop scoliosis. They actually have breathing issues as well. They are underweight. and then they develop contractures and then do not survive beyond 8 to 12 weeks, depending on which lab you are in. So this particular study has now been completed, and what she was able to show is not only does the mouse have breathing issues, but these breathing issues are not only related to the chest wall muscles, but at the time just before the mouse is running into significant troubles, that there are also changes within the lung tissue itself. This is kind of unusual because it is thought in muscular dystrophy that the breathing issues are only related to the chest wall, scoliosis, to weakness and muscle involvement, and not to actual involvement of the lung tissue. And so it really begs the question whether this is just something that is found in the mouse or whether this is also happening in individuals, human beings, who have LAMA2 CMD. She also did a pilot study of losartan, where she gave the DYW mouse losartan, which had been shown in 2 other labs to have a beneficial effect. Losartan is a medication that is commonly used to treat high blood pressure both in children and in adults. Therefore is a drug that already has a pediatric dosing, though it is unclear whether the dose to treat blood pressure is in fact the same dose to treat LAMA2 CMD. And what she found, and she confirmed the results in the other 2 labs, that when one gives losartan, the mouse has an improvement in their muscle tissue and in survival. She currently is testing another combination therapy of losartan and human growth hormone, and those results are pending. That work is still in progress. She has also started to do work looking at biomarkers in the mouse in both the muscle and in serum or blood. So this goes back to Cecilia's question. Can you find a marker in the blood in the mouse or in the muscle in the mouse that changes when you give a treatment, and then do those same markers, are they the same that you can find in humans, so that you could actually show in a clinical trial that in fact your target, the aspect of the underlying disease that you are targeting with the drug has changed for the better. She also has partnered with another scientist to perform magnetic resonance imaging, or MRI, in the muscle of mice that are treated and not treated with losartan. So you can imagine doing an MRI on a mouse. It's kind of a funny picture, but no pun intended. But that is what she currently is engaged in. So that has been Sweta's work. Does anybody have any questions?
I just have one question. Have a lot of kids started, have any kids started going on the losartan?
It's a good question. There are 2 kids who have started going on the losartan. The issue is we don't know what the right dose is, and the more kids who go on losartan, the less likely we will be able to have a clinical trial of losartan, which is what we're trying to get to, because the only way that you could really prove that the drug works is to do a clinical trial.
Yeah.
And this is the challenge. The drug is available, it's accessible, but you know, you also don't want to give a drug where you're not sure whether it's actually doing something. And so that's the real challenge here, and that is really our goal, is to get to a clinical trial with losartan in LAMA2 CMD. So that's really one of our goals in the next year and a half. So this is just to illustrate how scientists in the past used to think about muscular dystrophy. So the way they used to think about it is that there would be a loss of a key protein that was at the surface of the muscle cell membrane, and that then when the muscle was contracting, that because of this missing protein, that muscle membrane was not stable, and therefore it led to changes in calcium levels, rupture of that membrane, and muscle cell death, and that then that over time led to a loss of muscle muscle cells, which we call degeneration, and even if the muscle tried to regenerate and make itself new, that it wasn't able to keep up. And so cycle after cycle of this muscle membrane rupturing and the muscle not being able to keep up with regeneration is what in fact led to progressive muscle weakness, muscle loss, and muscular dystrophy as we know it in our families. What has really been fantastic is in part because this mouse model, the DYW mouse model, is such a good mouse model of LAMA2 CMD, and because it is severely affected, it shows weakness. There have been many publications that have sought to try and tease out, is that really the only thing that is going on in this particular disease? And so thanks to the work of many, many scientists who've been very dedicated, many scientists have, or several have really dedicated themselves to LAMA2 CMD, they have established that really the problem here is not solely that this protein, laminin 211, or Merosin, is missing, but that this primary problem of that protein not being there leads to many signaling pathways that over time augment each other and lead to this problem of the matrix kind of, which is the area outside the muscle cell, being completely abnormal and muscle loss. The reason I'm bringing up this complicated slide is that when you start to think about developing treatments, you can think about treating the primary problem, which is that you don't have the protein. So you can say a primary therapy would be to try and put laminin 211 back into humans, or its twin sister, laminin 111. And the second approach will be to try and prevent these signaling pathways that are leading to further muscle loss and muscle destruction. So Omigapil would fit into that second category. It is a blocker of cell signaling. It is not treating the primary problem that kids with LAMA2 CMD do not have or have only partially functional laminin 211 or Merosin. It is not targeting that. It is targeting the downstream signaling. Okay? The other thing that has become more appreciated is that there is actually, this problem doesn't just start after birth, it is starting in utero. So that the absence of Merosin or laminin 211 at this muscle cell membrane surface is already leading to issues with the development of muscle from a very early time period. So this, of course, makes us think about how can we invest as an advocacy organization in trying to improve our understanding of what are real targets for LAMA2 CMD. If we target a primary replacement versus a downstream target, what is the size of the impact that it has on the mouse, let alone humans, and how can we push to recognize LAMA2 CMD at an earlier and earlier time point to potentially in the future make drugs available as early as possible in order to really target somebody directly around and after birth. Now, I'm saying this, and as you know, I have a daughter I explained who's 16 years old. So I remember when I first heard somebody saying these kind of words that it made me feel very sad because my daughter is already older, and I said, well, what chance will she have of participating in a clinical trial and getting the benefits of a future treatment? And it is true, as our children get older, and if there are any affected individuals on the phone who are older, you know, initially, most likely clinical trials will go in younger children because you have the greatest chance of potentially showing a bigger effect size. But then once a drug has been shown to be effective, then it will be made available for the entire community. Mm-hmm. Any questions?
How come they're not able to replicate the protein, like in some sort of form? So to, I don't know, inject it or putting it in a medication or something?
Yeah. So it's a great question, and it makes the most sense to all of us that, gosh, if we could just put that protein back there, this would be the best possible solution. So here are some of the reasons why that becomes problematic. There are some people with LAMA2 CMD who make zero protein. Okay, if you give them a protein that their body has never ever seen before, they will mount an immune reaction.
Mm-hmm.
Some kids with LAMA2 CMD make some laminin 211 or Merosin. They make some of the protein, it just isn't really functional. So those individuals could potentially benefit from, as you're saying, getting an injection of laminin 211. But the problem is how do you get that laminin 211 to the muscle where it needs to go? So, you know, when you inject something in the bloodstream, there are cells that will come, will try to recognize what that protein is. How will it know to direct itself like a traffic cop to the area of the body that it needs to go? And then when you think of how much of our body is actually composed of muscle, this is a really big problem. You need that protein to go everywhere to all the muscles. And that has been one of the hardest issues in terms of injecting small proteins, is figuring out how you can flag them so that they go where you need them to go in the body.
Mm-hmm. Yeah.
So one of the strategies that we're going to talk about next, laminin 111, is in fact that idea. So when a baby is in utero, it actually makes a different form of laminin called laminin 111. And shortly after birth, this laminin 111 production is shut off. And typically in a kid who is unaffected, they're making laminin 211 or Merosin in utero, and then they continue to make the laminin 211 after birth. So the idea behind developing laminin 111 therapy is that it is a very similar protein, but not exactly the same as laminin 211. And because it is so similar and somewhat of a fraternal twin, you could say, not identical, the idea was, can we actually produce it, and can we inject it, and do we see an impact if we inject it? And that is really the work of Dean Bourkin, who I will come to next. The other big problem in developing protein-based therapies is you need to be able to develop a protein that is not only stable so that when you inject it, it doesn't degrade, that it actually can tolerate the pH or the acidity level of the blood. And then number two, that you actually have a way of making that protein. You know, you can't just all of a sudden say, hey, I want this protein, let's build a factory and make this protein. It's actually very complicated to make proteins, and some proteins, because of their structure, cannot be made in a synthetic way. Okay?
Thank you.
Great questions. Okay, so just to show you the results of the funded research, so as a result of the Cure CMD funding, Sveta Gergenrath has been able to publish multiple publications that really help us have a better understanding of LAMA2 CMD, and in particular, her real contribution to the field has been this idea of combination therapy. So the next steps are to model what, if you treat a mouse with losartan, what does the blood and the muscle look like? Are there markers that respond to treatment with losartan? And is there perhaps a drug that is losartan but is not losartan that might even be better than losartan? Because losartan, again, is not a cure. It is not a primary replacement of the missing protein. It is targeting the downstream signaling. The other steps that we are taking, and when I say we, I mean Cure CMD, is that we are engaging with the pharmaceutical sector to ask these very questions. Are there drugs that are like losartan that you have on your shelf that you might be interested in thinking about using in LAMA2 CMD? So it's this process of building relationships with pharmaceutical companies in order to start to increase their awareness and their interest in contributing and moving forward in CMD. And then the last next step that we have is an idea of launching a toddler natural history study. So this gets to the question that somebody had raised about the age for the Omigapil PK study. And what we're really interested in defining is, are there endpoints in young children with LAMA2 CMD? So in children who cannot do pulmonary function tests, are there other things that you could measure, including blood levels, that might actually give us something that we could use for a clinical trial in the younger kids? Okay, so those are some of the plans that we have. We Cure CMD in the next year. So this is Dr. Dean Birkin, who's at the University of Nevada, and Dr. Birkin has also been very interested and pursued LAMA2 CMD very intensely. He first tested laminin 111, and we funded his research to test laminin 111, the mouse version of it, in the DYW mouse model. And in his study, he showed that when you delivered this to the mouse model, it improved strength and survival, and it also improved the ability for the muscle cells to regenerate. He is currently funded to test not only the mouse but also the human laminin 111 in the DYW mouse model as kind of a pre-step, as part of the steps that you need to check off in order to move laminin 111 forward into clinical trials. And those results are pending. In this particular study, he also looked at biomarkers in the muscle and in the blood in order to see whether, when one treats someone with laminin 111, that one sees a change at the cell and the blood level. So results of his funded research, he's published 2 different papers that were made possible through this funding. And then this fall, he received a very large grant from the National Institutes of Health using the data that came from this funded research to apply for additional funding to move forward additional steps to test laminin 111 in the DYW mouse model, all part of this preclinical testing that is needed before one moves forward towards clinical trials. As many of you know, Alexion is a biotech company that has acquired the license for laminin 111. And they are currently working to replicate the results that were obtained in the Birkin lab in order to determine whether they would like to move forward with laminin 111 in future CMD clinical trials. So we have continued to engage with Alexion. One of the big issues is, of course, this registration of the global population. So as we're thinking through the steps that will need to be addressed in order to get to clinical trials, we recognize that we need to simply find more individuals with LAMA2 CMD. And in many individuals, they may have had one type of genetic testing where one mutation was found, but the other mutation was not found, in part because deletion and duplication testing was not performed in those individuals. So one of the other things that we are looking at is acquiring sponsorship to provide free deletion duplication testing to additional individuals who have had only one of their variants identified, one of their mutations found, because for future clinical trials it will be very important that people have both mutations found in order to confirm that genetically that they have LAMA2 CMD. And then we are finalizing the results on the CMD-Com natural history study to identify the clinical trial endpoints that could be used for a future clinical trial. So this is Dr. Olga Igucheva, who, together with Dr. Monli Chu, I couldn't find a picture of her, from Thomas Jefferson University, has been funded by Cure CMD to also look into a very unique way of replacing the missing protein. So it turns out that certain cells, probably about 10-15 minutes, certain cells called human adipose stem cells actually are little protein factories, and they make human laminin 211. They also make collagen 6. And so her idea was, I wonder if we could inject these adipose stem cells into mice and see if they will continue to produce laminin 211 if they are injected. So it is a stem cell approach, but it is not stem cells to create muscle. It is stem cells to act as protein factories to create the missing protein that is not currently available in the muscle. So she, during the first phase of her funded research, was able to show that she was able to inject these cells, that once they were in the muscle, they were able to spread throughout the muscle, and that once they were in the muscle, they continued to produce this laminin 211. So she has been funded again now to answer the pressing question of, is there a way that she can identify certain populations of these cells that are predisposed to going to muscle so that she can actually increase the number of human adipose stem cells that would actually go to muscle and preferentially target muscle so that she can get those cells into the muscle if they were injected in an IV and thus have the protein being produced in the muscle that is missing. And those results are pending. So just to recap, two of the scientists are working on a primary protein replacement strategy with either laminin 111 or laminin 211, and one scientist is working on blocking downstream signaling pathways. There are, besides these three scientists who Cure CMD has funded, additional scientists who I have not highlighted who have also dedicated much time and energy towards LAMA2 CMD. And perhaps on a future call we could have a scientist join us and also review the work of additional scientists who have been working on LAMA2 CMD. So one of the other areas that we have invested in heavily is in pursuing biomarkers. So biomarkers are signals in muscle or in blood or in on imaging that can tell us how the disease is doing. Is the disease getting worse? Is it getting better? What's going on? The reason it's important to have biomarkers is that in a clinical trial, especially in a disease that is slow-moving, as LAMA2 CMD is, and there are also periods in LAMA2 CMD where there is great stability. So typically between ages 5 and 10, kids actually seem to improve somewhat and have a period of stability. So how can you then, when you give a drug, actually know whether the drug is causing an improvement or this improvement is just part of the natural course of LAMA2 CMD? So one of the best ways to be able to do this is to have a biomarker, something that changes over time in the disease, and in particular a biomarker, something that you could say, when I give this drug, I know it targets this pathway, and this biomarker sits on that pathway, and that's how I can monitor whether there's actually an improvement in the disease and whether my drug is actually targeting what I think it is. So we early on, as part of the CMD natural history study, there has been blood drawn in order to evaluate a protein signal in the blood that changes over time with age and with changes in muscle strength. So Dr. Jim Collins received a grant from Cure CMD to continue to do that work. That work is still pending as the analysis is ongoing from this 5-year natural history study. As part of the CMD natural history study, Dr. Bonneman, who is the principal investigator for the natural history study, also has performed MRI in individuals with LAMA2 CMD over time to see whether, in fact, you could see a change in the signal in the MRI of muscles over time that could also be used to say whether a drug is having an effect. And then the other investigators, Dr. Russell Butterfield and Professor Montoni, have received funding to look both in cell lines as well as in blood and muscle in humans affected with LAMA2 CMD in kids, to see whether there is in fact a signal that could tell us how the disease is progressing over time. So those results are pending. So just to conclude, what are our priorities for funding for 2015? So I have spoken about number one, the ways that you can contribute to moving the field forward by registering, by trying to identify individuals in your community who may not have registered, by sending in your medical records, by participating in clinical studies and trials if you are eligible, and by participating in webinars such as today's webinar to learn more about what is going on in our community and how can you work together with the community to affect change. I also want to point out that on the CMDIR and Cure CMD websites, there are many resources regarding care and management, including consensus guidelines and family guides that have been translated into multiple different languages, as well as videos that you can watch that can provide you with additional information. And again, if you have any questions, I'm happy to answer any questions that you might have. So as we are thinking through Cure CMD, as we're thinking through our priorities for the next year, we really would like to ensure that we have a genetically confirmed pool of people with LAMA2 CMD who will be ready for clinical trials. And as I had mentioned, there are a sufficient number of families who have, where the child has only one mutation identified, not those mutations identified. Identifying a sponsor to provide additional genetic testing to those individuals would really go a long way for us to ensure that we have the large number of people who are genetically confirmed. We also will likely need to extend this hyperinsufflation trial in the United States in order to recruit these additional individuals, and we anticipate that the cost of extending the hyperinsufflation trial will cost about $1,000. We will be receiving a grant application for biglycan, which is a novel linking molecule that has been in human cell lines with LAMA2 CMD, and in those human cell lines it has shown an improvement in several of the signaling pathways in those human cell lines. So we will be receiving that application, and we are likely to receive additional research applications on January 15th. So as part of our annual research grant application process, we typically receive anywhere between 3 and 6 grants that are requesting funding to move forward LAMA2 CMD science. And those grants typically are funded anywhere between $25,000 to $45,000 on an annual basis. So these are ongoing kind of funding needs in order to move the LAMA2 CMD field forward. The Omigapil PK study, currently Cure CMD has set aside $100K in funding and has identified additional funding partners. However, if there is a need to extend the PK study because of enrollment issues, this may come up as an additional funding priority. And then finally, as I had mentioned, we are interested in moving forward a toddler LAMA2 natural history study and anticipate that the cost for running that study will be somewhere around $45,000. So that in short is a kind of quick update on the registry, current clinical trials, and research that is being conducted, as well as an update in terms of our priorities moving forward for 2015 for LAMA2 CMD. And I'd be happy to take any additional questions that anybody has, and can also answer questions offline. Feel free to email me. My email address is Anne, A-N-N-E, Rutkowski, R-U-T-K-O-W-S-K-I, at curecmd.org. So I'm happy to take any questions. And Pat, if you have any additional thoughts that you would like to share as well, this would be wonderful if you wanted to share some of your thoughts.
Yeah, no, that's great. I certainly don't have anything to add over and above walking through the research, what you did, and want to thank you again for walking us all through it. It's an awful lot to know and understand, and certainly speak for everybody as far as all the appreciation for you doing that and breaking it down for a lot of us who aren't scientists or doctors. It's just helpful. So I'd also like to say to everybody, by all means, if you have any questions or thoughts Cure CMD related, maybe not so much on the research side, because Anna is certainly better than I am at explaining that. But you can always feel free to reach out to me as well. My email address is pat.may, so P-A-T .may, like the month, at curecmd.org. So.
Yeah, I'm Valérie from France. There was a trial on doxycycline. There was still ongoing, or it's finished?
Yes. So, Valerie, it's wonderful to have you on the call. Thank you for calling in. Dr. Sweta Gergenrath performed a study comparing doxycycline and minocycline, two antibiotics that have, in addition to how we think of them as antibiotics, have anti-inflammatory properties and anti-cell death properties, kind of in the vein of Omigapil. And she was able to show that doxycycline did show an improvement in the mouse model. There has not been anybody who has decided to do a clinical trial with doxycycline in humans. So there is no data on whether or not this drug, which is, again, readily available, though it is not used in children because of side effect profiles, but in adults, there's been no thought of doing a clinical trial with doxycycline. So currently all we have are her results in the mouse model.
Okay, thanks.
You're welcome. Valerie does bring up a good point, and I will just throw this out there, that in older individuals who are thinking, okay, it may be some time before a clinical trial happens that AI can participate in, that in some individuals they do ask themselves the question, are there drugs that are already available, that are off-label, that have been shown in the mouse model to have a benefit, and what is the risk to me if I take them, not knowing what the correct dose is, and what is the potential benefit, and do I want to do a trial on myself as one person to just see, do I see a positive benefit? If you do something like that, it is important to have a physical therapist who can measure some measure of muscle strength or breathing function so that you have an objective answer to your question of, does this particular drug show any positive effect? The one other thing one has to always keep in mind as well is that there is a very strong placebo effect, meaning even if you were to take a sugar pill instead of an actual drug, the mere fact of taking the sugar pill can make you feel better 20% of the time. So I think the one thing that I would like to say is that if somebody decides that that is something that they would like to do, first I would love to know about it because it helps me understand whether in fact people have appreciated a change. Even if there's a change in their management, for example, if people decide that they're going to do the cough assist on a regular basis or they are making a change in their ventilatory management, it's very helpful for me to actually know this because then it helps me kind of think through, is this a strategy that needs to move forward into a clinical trial, and do we need to prioritize it because of some positive results, even if in individuals who are either doing this on their own with their physician or in a group of individuals, such as the individuals in France, for example, for hyperinsufflation, who are under the care of Dr. Susanna Quijano-Roy. I know this was a lot of information to process. We have recorded this. And what we will do is we will send out through the CMDIR a link to this video recording so that you can listen to it again on your own. And if you have additional questions that come up, you can email me or email Pat, and we're happy to answer any questions that you might have. Well, first I would like to thank everybody for taking the time to be on the call today. It really was wonderful to hear all of your voices and to connect, even though we are living in places not so close to one another. But it always makes the community feel smaller, and I really want to thank everybody for taking the time today. And then I look forward to hearing from you if you have questions, and we will let you know when we plan on scheduling the next call. It would be very helpful to hear from you too whether you thought that this call was useful and provided you with information that you wanted to hear, and if there are additional topics that you would like to see addressed on future calls. And I wish you all a pleasant rest of your day or evening.